Dear scholars, in this video I will discuss about drug development process. There are basically four stages of drug development process. Stage 1 discovery and development, stage 2 preclinical research, stage 3 clinical development, and stage 4 FDA review and approval. Drug discovery can be defined as the process to bring new pharmaceutical drug into the market once lead compound has been identified. There are basically 4 points in the first stage of drug discovery which you should understand properly, and they are target selection, target validation, lead generation, and lead optimization. In target identification, researchers try to find biological target like receptor, enzyme, protein, DNA, etc., which are basically involved in disease process. If I take a very recent example of COVID-19, COVID-19 attaches with the angiotensin converting enzyme 2 receptors. That's why some doctors or hospitals use ACE inhibitors to treat COVID patients. It is the process to identify if the target molecule involved in disease process. Target validation ensures that engagement of target has potential therapeutic benefit. Lead generation or lead identification. It is also known as hit to lead, and this is a process to identify the compound that can interact with the target protein or target receptors and produce significant activity. Lead optimization. Lead optimization is the process to synthesize and modify to improve the potency and reduce the side effect of newly discovered drug. In stage 2, preclinical studies are performed on animals, and preclinical studies are subject to the FDA regulations. In India, main regulatory bodies are Committee for the Purpose of Control and Supervision of Experiments on Animal and Institutional Animal Ethics Committee. You can note down the functions and role of CPCS, EA and IAEC. We know that preclinical studies are performed on animals, and the main goals of preclinical studies are to determine a starting and safe dose and assess potential toxicity of the product. In preclinical studies, elevated dose of drug is tested using in vitro, means either in test tube or cell culture, and in vivo, means on animal. The main point researchers determine are pharmacokinetic parameters like absorption, distribution, metabolization, and excretion, and pharmacodynamics of drug like benefits and mechanism of action and best doses and administration route, side effects, adverse events, and effectiveness of the novel drug compared to the similar existing drugs. If you enrolled in advanced course like M.Pharm and PharmD, you need to study a bit deeper. There are two approaches of preclinical studies. First one is a pharmacological approach and second one is a toxicological approach. The pharmacological studies are categorized into two groups: research pharmacology studies and safety pharmacology studies. And then research pharmacology studies are further categorized into two groups: primary research pharmacology studies and secondary research pharmacology studies. The primary research pharmacological studies are focused on mechanism of action of drug, and the study in vitro focus on the drug action on specific receptor sites. And the studies conducted in vivo investigate the pharmacological action of the drug in animal models. The secondary pharmacological studies focus on the overall pharmacological activity of the drug. In simple terms, this study focuses on other therapeutic effects of the novel drug and also check if the novel drug binds with other receptors apart from the target receptors. The safety pharmacological studies that focus and investigate potential undesirable effects of the novel drug. Dear scholars, you can note down the main point of the toxicological studies mentioned on the screen. Once preclinical studies are completed, sponsor or developer need to submit investigational new drug application to FDA to get approval for clinical trials. If FDA approves investigational new drug application, then researchers can move for the stage 3 I mean clinical research process. The goal of clinical trial is to determine if a new drug is effective to treat disease safely. There are basically three phases of clinical trials which determine if the new drug is safe for human use. In addition, phase 0 is added, where 10 to 15 people picked and given subtherapeutic dose or microdoses. The phase 1 clinical trial tests the safety, side effects, and best dose. In phase 1 trials, number of participants will be around 20 to 100 healthy volunteers. The length of phase 1 study is about several months. Approximately 70% of drugs move to next phase trials. Please note that the single ascending dose and multiple ascending dose are the very important part of the phase 1 clinical trials. A phase 2 clinical trial is conducted to evaluate the effectiveness and safety of the new drug. The phase 2 study is performed on 100 to 300 people with the disease, and the length of study is up to 2 years. Approximately 33% of drug move to the next phase. Phase 3 clinical trial compares the safety and effectiveness of the new treatment against the current standard treatment, because physicians do not know which treatment is better. Phase 3 trials involve thousands of patients and further assess safety and monitoring of side effects. Length of phase 3 study is 1 to 4 years, and approximately 30% of drugs move to the next phase. After the successful of phase 3 trials, new drug application sent to the FDA for review and approval. After successful phase 3 trials, data collected in clinical trials is sent to the FDA for review and approval. The FDA looks at all the data from the 3 phases of clinical trial to determine if the investigational product should be sold to the customer. Once FDA receives a new drug application, if the new drug application is complete, the review team has take 6 to 10 months to make decision on whether to approve drug or not. On the screen you can see NDA review timeline. Once FDA approves new drug application, manufacturer or sponsor can proceed to post-marketing survey. Phase 4 trials are conducted to determine long-term safety and effectiveness of drug. It is mainly to identify adverse effects of the drug that could not detected in previous studies. Thanks for watching. Please subscribe, like and drop comments to support my work.